Hello everybody, today we are going to discuss cholinergic neurons. We will discuss how cholinergic neurons synthesize the neurotransmitter and how cholinergic neurons secrete the neurotransmitter and how neurotransmitter will bind to its respective receptors. So basically, we categorize the neurons on the basis of the neurotransmitter. Those neurons that secrete the acetylcholine neurotransmitters are called cholinergic neurons, and those neurons that secrete the norepinephrine neurotransmitter, we have categorized these types of the neurons into the adrenergic neurons. So we will see how cholinergic neurons synthesize the acetylcholine. So let's get started. First of all, here is the terminal end of the cholinergic neurons. Choline, which is the precursor substance of the acetylcholine, is present into the extracellular space. So, choline is present in the extracellular space. Here is the choline. And sodium also present in the extracellular space. Now, choline will transport into the neuron with the help of the co-transporter. Now, these co-transporter will utilize the ATP and transport the choline into the neurons. Now, with the help of the co-transporter, this choline will enter into the neurons along with the sodium ion. This is the important thing. Now, after entrance into the neuron, this choline will bind to the acetyl-CoA enzyme A. This is the important thing. This will bind to the acetyl-CoA enzyme A, or you can write it with the acetylCoA, acetyl coenzyme A. Now this acetyl coenzyme A and choline undergo reactions and convert into the acetylcholine. Acetylcholine. Or you can write it with the ACH. Acetylcholine is represented by the ACH. So reaction between the acetyl coenzyme A and the choline, and in return for that you will see the synthesis of the acetylcholine. The most important enzyme that is used in this reaction is the acetyltransferase. Acetyltransferase enzyme. Acetyltransferase enzyme. That is the important thing. Choline enter into the neurons, and after the reactions you will see the synthesis of the acetylcholine. Now, this choline can be inhibited by the certain drugs, and that drug is the hemicolinium. Hemicolinium is a drug that blocks the entrance of the choline into the neuron. When there is no entrance, there is no synthesis of the acetylcholine, which is a neurotransmitter. Acetylcholine is a charged neurotransmitter. It has charge on it. If we look at the structure of the acetylcholine, so you will see the presence of the nitrogen, and there is a positive charge on the nitrogen, and along with that you will see the CH2, CH2O, C double bond O, and CH3. This is the structure of the acetylcholine. Now, if you look, acetylcholine has a positive charge on it. Okay. Now we know that the synaptic vesicle is present inside the neurons. For the entrance of the acetylcholine into the vesicle, you require the ATP because it has charge, and the charged particle mostly require ATP for its transportation from one place to another. Now this acetylcholine will enter into the vesicles by using the energy because it's a charged particle. So acetylcholine has been entered into the vesicles, and lot of other things will also be present in the vesicle, that is proteoglycan and the ATP. So these are the important things that are actually present in the vesicle, presynaptic vesicles. This vesicle prevent the acetylcholine from degradations by the different enzymes. Now this vesicle starts moving towards the last membrane, that is presynaptic membrane of the neuron. This one is the presynaptic membrane of the neuron, and this vesicle fuse with the presynaptic membrane, and ultimately acetylcholine will start releasing into the synaptic space or the effector sites. Now movement of the vesicle to the presynaptic membrane, this will happen. There is another important thing, as we know that the electrical impulse transmit through the neuron. When the electrical impulse reaches at the terminal site of the neurons, lot of calcium starts entering into the neurons, calcium ions. Now this calcium ion, increase in calcium ion promote the fusion of the presynaptic vesicle to the synaptic membrane, and ultimately you will see acetylcholine start releasing into the space. That is the important thing. But important thing, release of the acetylcholine can be regulated by different factors. At the presynaptic membrane, you will see the receptor, that is presynaptic receptor. When you have to stop the release of the acetylcholine, this acetylcholine bind to its receptor that is present on the presynaptic membrane and stops the release of the acetylcholine. This is the normal process. This will happen through the feedback mechanism. Increase in acetyl concentration, when we have to stop the secretion of the acetylcholine, then acetylcholine bind to the receptor and ultimately stop the release of the acetylcholine into the space. But there are certain conditions that can enhance or that can inhibit the release of the acetylcholine. For example, botulinum toxin produced by the bacteria Clostridium. This toxin inhibit the release of the acetylcholine. When there is no acetylcholine, then there is no signal transduction to the other neurons or to the effector side. And the most important is the neuromuscular junction. When there is no signal transduction to the neuromuscular junction, then there is no control of the muscles under the nervous system. Ultimately, you will see the paralysis in that case. Next is the spider venom. This spider venom increase the release of the acetylcholine from the vesicle. So botulinum inhibit the release of the acetylcholine, and the spider venom increase the or enhance the release of the acetylcholine. Okay, these are the important things regarding the release of the acetylcholine. Now, this acetylcholine can bind to the receptors. Two main receptors through which the acetylcholine bind. First one is the muscarinic receptor. And second is the nicotinic receptor. These are the two important receptors to which the acetylcholine bind. Nicotinic receptors mostly present on the neurons and at the neuromuscular junction site. But the muscarinic receptor mostly present on the different organs that acts as effector. When the acetylcholine bind to the nicotinic receptor, then ultimately signal transduct inside the cell. And as a result, you will see the process, and that is ionotrophic ionotropic process, you will see the signal transduction by the exchange of the ion through the cell membrane. So this is how acetylcholine influence the nicotinic receptor, and this is how signal transduct inside the cell through the ionotropic mechanism. When the acetylcholine bind to the muscarinic receptor, then as a result you will see the different scenario, and in that case you will see the metatrophic process, and as a result you will see two mechanisms. One is the adenylyl cyclase mechanism, and second is the phospholipase C mechanism. So signal transduct through the muscarinic receptor through two mechanisms. First is the adenylyl cyclase and second is the phospholipase C. So this is how acetylcholine binds to the nicotinic receptor and the muscarinic receptor. These are present on the neurons and the neuromuscular junction, and this is present on the muscarinic receptor. Now, this acetylcholine breaks into the two components. First one is the choline and second one is the acetate. This degradations of the acetylcholines happens in the presence of the enzyme, that is acetylcholinesterase enzyme. Acetylcholinesterase enzyme. This enzyme degrade the acetylcholine into the choline and the acetate. Now this choline again utilized in the synthesis of the acetylcholine. So this is how acetylcholine degrade, and again choline is used in the synthesis of the acetylcholine. So there are two category of the drugs. First one is the cholinergic agonist and second is the cholinergic antagonist. Cholinergic agonist drugs act similar to the acetylcholine, but the cholinergic agonist drugs works opposite to the acetylcholine. So this is all about the cholinergic neuron. If you still have any question, you may ask in the comment section. Thank you so much.